Okay, so fix. Okay, that's a tool, the goal of that tool, so I will start with the goal. So, for example, you have antigen sequence, and you can model the sequence in the structure. You have antibody sequence. You again can build a model, and then you can proceed with the docking with the goal to understand the discontinuous epitope and how antibody and antigen interact. So, and there is some other parts of that computational flow which I won't discuss. So, when in that case, when you have a sequence and you need a structure, you can use a structure of a protein either from PPT or you can look at protein modeling profile that we already discussed. For the docking, there are two servers which I will show you and recommend to use ClassPro and SageDoc. They will show you how they work. So, and this part is covered with this. So there are some other approaches, Rosetta antibody and web antibody modeling, which allow to do the same, which allow to build a model for the antibody sequence. So, and we did kind of some research because we wanted to put something useful on our website, and we figured out that Rosetta antibody is very slow and takes a lot of CPU, and the web antibody modeling is slow forever. So you can wait for, I waited for 2 weeks probably, and the results. So, and we decided on 6, and actually nothing else is available, by the way. So, we implemented 6 on our website, and now you can use it. So, it's available here in B-cell epitope prediction tool. Well, it's not exactly for B-cell epitope prediction, it's for antibody modeling. So, but we decided not to go with many tabs for different kinds of tools, so we put it here. And the tool was designed, it was published in 2008, and it was developed in Italy in the Anna Tramontana group. And they shared the code with us, so we could put it on our website. So, and that's how the tool works. So, you can start with trying it using example because it will be fast, because example is already cached. You need to submit 2 sequences, one sequence for light chain antibody and for heavy chain, and then click submit, and that will be the result. So the result will be in the, again, JMOL viewer will open, and what you can do, you can select CDR regions, which will be in different colors. You can start a data structure and to see, for example, that like H3 CDR region is very close to light chain, while those H1 and H2 are kind of outward. So you can just play rotating the structure, and again it's JMOL, and you can use all options which are on the right button of mouse to play with JMOL. So, but the most interesting part comes when you click download model file. So when you download, you will get PPT file with a summary mark which provides the alignment for heavy and light chain, alignment of your submitted sequences to the template in the database which supports the tool. And further, you scrolling down, you will see just the standard format for PPT file with atom coordinates for each atom provided. So, and that file will have extension .pdb, and now you can use that file in different applications. So, and I will show you an example which I did. So, I looked at different antibodies which are on market, and I selected this one, tocilizumab. I think it's called tocilizumab. So it recognizes interleukin-6 receptor, and it's used for treatment of rheumatoid arthritis. So my goal was to take sequence. So I looked at the structures that was available for this antibody. So I needed to look for sequence. So where to get sequence? So sometimes, if you're on Wikipedia, sometimes you can find key information for sequence. But I was not so lucky. So so that's what we're now trying to do, trying to find the sequences and to model that antibody. So I went to the IMGT database, because IMGT database has a monoclonal antibody database, which actually has all therapeutic antibodies and provides all available information about them. So I went to that database, and I just typed here tocilizumab, click search, and I got the result, the page result of that antibody. So, and you immediately can see a lot of different information. So, but I was interested in sequences. So sequences will be here. So you click on kind of not obvious, but still. You click here, and you will get that standard representation for structures in IMGT, where you will get a sequence for heavy chain and below for light chain, and you can get sequence in FASTA format. So I click sequence in FASTA format. I got a new window open with the sequence, and then I got sequence for the light chain. So I scroll down, sequence for the light chain. That's my sequence. So then I took all those sequences, I trimmed all unnecessary characters, and I just cut and paste them in the tool and click submit. So, and the tool just took 2 seconds to get me the result. So that's my model of the tocilizumab antibody. So now I can download that model, and I have a model for antibody. Now I need to solve the problem for interleukin. So if there is a structure, or I need to practice with the modeling. So I look for UniProt for interleukin-6 human receptors, and scrolling down in UniProt, you can see, because you have no idea, when you hear, you have no idea about species. For BDD, it doesn't show you species, so it can be just homolog. So, and it's more convenient to go to protein model portal directly. So I went here, and what I saw that there are 4 models which cover different parts of the protein, but not the whole protein. Okay, I selected one which covers the largest part of the protein, and that's exact structure. And it's actually a structure from human interleukin, so it's nice. So I probably don't want to do any more than it. I just can take that structure. So I clicked here, and I went to PDB. And then on the right, I click download file, and I click PDB file as a text to download, and I got 2 files. So that's my model, and that's PDB file. So now I can go and use docking tool using those 2 files and try to dock models antibody to existing structures interleukin. So now I go for that part, antibody antigen docking. So again, I will show you how I play with these 2 orders. So this is page doc. So and so I submitted a receptor, it will be my antibody. So I submitted the model, which I have, and here because they allow to select PDB, chain, and ID. So I just typed my PDB chain and ID. Otherwise, I could submit a file. So, and they require email. And now you need not to forget to select the mode which you are interested in, because they provide different modes. And for antibody antigen mode, they will be looking for docking mode, which involves only CDI region. So they filter out all other models. So when you click submit, and PageDoc is very fast. You just get results in a couple minutes, but on your email. But when you click on the link, you will get on that page. And here you can download, again, a PDB file. Here you can download any model. It will be a PDB file which will contain both antibody and protein. And from that file, you can calculate your epitope, basically, the residues which interact with antibody. There is also a new function which they added very recently, which is called JMO view. If you go here, you can highlight any model predicted, and it will be shown in JMO. Then you can select several structures and zoom in, zoom out, and to see like where antibody binds. So in that case, all three top structures, they bind in a kind of similar way, but it's hard to tell, and unfortunately, they do not provide you option to analyze it further. So you need to find a way now to download files and do analysis on your own. So another tool is Class Pro. And again, here you can either submit a file or select CDB. So I submitted for antibody, submitted a file. For protein, I just submitted, provided CDB and chain. And now you need to use advanced options before to proceed. And here are advanced options. So, and you need to select antibody mode. So highlight both. And why? Because people who developed the Class Pro, they specifically train the scoring function, voting scoring function, on antibody protein complex. So, and it's actually very good, very well working on benchmark. So, and then you can do doc. And then it will start running, and depending on how quickly they accrue, it can take several hours. It took me about 3 hours to wait for the result. So, and then you can display up to 50 predictions, and you can download all models. But here on the website page, which will show up after calculation, you just can fast look at where antibodies, the antibody binds protein, kind of in different places, in different places. So that means that it's very hard to recommend which model to select. The first one, the second, then best, so, and that's the nature of working algorithm. Models or energy? So yeah, they give. They should give the energy. There is a view model scores here. So they will give you scores. So and you can select models based on those scores. Okay. Yes. Is this practice PCR antigen fraction or an MSC? Okay, yes. The question is if that the same strategy can be used for docking PCR to peptide MHC complexes or peptide to MHC. Okay, I personally don't know about available servers which specifically designed for that stuff. You can try to use like those tools as a general process for the project. But you said something about the training basically for that. Yes, here they train, but I don't know exactly about about the tools which been trained for both in peptide to MHC and to both PCR to peptide MHC. So there are some works published, but I don't know about further. Yes. So the question is how it works.
Yeah.
Okay. I can recommend you to look at the paper for details. But yes, it works based on homology model. I mean, in general. But there are some details where they account for non-conformation for CDR conditions. Yes, the question was if there were any benchmarks for PICS. Yeah, they published it in their paper. So you can click on the reference on the tab and you will see the table. I wanted to show that, but somehow decided not to. There is a table which provides the results of evaluation of how it works for a new antibody structure.
My last question. If I have like I have maybe not do it one by one. Then
okay, the question was if any paralyzed version exists for modeling antibodies, yes, on the large scale. I don't know of any. So, PICS, do you provide PICS downloadable version?
Epitope is downloadable. Epitope is downloadable.
So, no, we don't, yes. Because if some software you can download it as executable, like Rosetta Stone, then you can organize it by yourself just to go through. But Rosetta Stone is very slow, by the way. And it can take, like for, I don't know, for 100 structures, it will take you a month probably for today. Yeah. And PICS, yeah, you can contact the authors and ask if we can provide executable, and you can give us that suggestion. We might do that eventually. Yes?
great.
Okay, thank you.